In this video, we will discuss about this pancreatic cancer cell stroma and what are the therapeutic targets that are used to disrupt this stroma at different sites. And after that, we will end this video with some brief overview about this cancer-associated fibroblast, how it will help towards this towards the aggressiveness of this pancreatic cancer. So this is the pancreatic tumors that is shown in the center with this blue color, and whatever you are seeing in surrounding these tumors are the result of stroma and the tumor microenvironment. So one of the essential thing that I have discussed in many times in our previous videos is the food supply for these tumors, and this one of the major way by which they get their food supply is through these blood vessels, or they will create more blood vessels, and which is known as angiogenesis. And this blood vessels, if we can somehow disrupt these blood vessels, so that tumor cells will get deprived of these nutrients and will finally die after some time. So there are therapeutic targets. There are therapeutics research that is going on to disrupt this angiogenesis, and these agents that disrupt this angiogenesis of the blood vessels towards this tumor is known as anti-angiogenic agents, and this will disrupt the food supply of these tumor cells and helps in disrupting the the modified metabolics in the tumor cells. The next target that they have used is to disrupt this extracellular matrix. The extracellular matrix act as a barrier, physical barrier, so that the immune cells will not be able to do, or this extracellular matrix prevents the immune surveillance from immune surveillance, so that the immune system will not be able to detect these tumor cells and act against them. So there are several commercial therapeutic targets are available to disrupt this extracellular matrix, and that are mentioned here. That is PG, PH20, and defectinib, and there are other targets that are mentioned here. You can refer them. And after that, the another important part of this stroma around the pancreatic cancers is the cancer associated fibroblast, and as this will act as a not this will not only just act as a barrier to this tumor act as a barrier between the immune cells and the tumor cells, but it will also help these tumor cells in the process of metastasis. So these things can also be disrupted using these commercially available therapeutic targets, that is vimtogenib. So vimtogenib will act not only on this cancer associated fibroblast, but it will also act on the other extracellular matrix component. So, and it also has a multiple effect when this vismodegib is able to overcome this extracellular matrix and cancer associated fibroblast. It can also attack on the tumor cells, and the vast research is presently going on is to uh, enhancing the immune cells by providing additional information to the immune cells about the tumor cells, so that they can effectively target the tumor cells, and helps this additional information will also help the immune cells to distinguish them from the to distinguish them between the tumor cells and also the healthy cells. So such as this CAR T cells therapy, where the receptors of the antigen that are specific to the tumor cells will be provided to these T cells, which is known as chimeric antigen receptors, so that they can target these tumor cells more efficiently, and don't need to spend time in understanding this antigen specific antigen on the tumors and develop a antigen later on. They can directly start generating antibodies against the antigens that has been provided by this CAR T cells, that is chimeric antigen receptor cells. And there are other immunotherapies are also mentioned here, which are used to enhance the immune system against the tumor cells. So this is just a brief overview how these different cell stroma component are being targeted by these different therapeutics agents, and the research are still going on to further improve the efficiency of these therapeutic targets and reduce the side effects. Now let us quickly go over this cancer associated fibroblast role in the pancreatic cancer. So this, as you know, that this pancreatic tumor is the result of desmoplasia reaction on the pancreatic cancer on the pancreatic organs, and because of that the tumor cells will be formed and they will start to interact with its neighboring cells, and that cause in the stroma formation and tumor microenvironment formation. And this cancer associated fibroblast is the part of this tumor microenvironment. So it will have several functions to support these tumor cells, to support the growth of these tumor cells. And one of the function is it helps in the crosstalk with the extracellular matrix. It acts as a intermediate between the tumor cells and the extracellular matrix. And also as we have already understand from the previous videos, it acts as a physical it acts as a one of the component of the physical barriers to prevent the immune surveillance of the tumor cells. And as it is shown here, it acts as a direct contact. It means that it acts as a physical barrier and prevents the physical barrier between the immune cells and the tumor cells. And it will also generate this inflammatory signals that will that's usual function is to support the immune system. However, this cancer associated fibroblast will produce immunitory signals in a way that will support the tumor growth, such as autoimmune response that will further support the tumor aggressiveness. And it will also cause this DNA damage and physiological stress such that these conditions will support tumor aggressiveness, such as hypoxia condition or DNA damage, in a way that the energy will be channelized towards the uncontrolled growth rate of the tumor cells. So, and we have already discussed some of the metabolic reprogram that occurs within the pancreatic tumors in our previous videos. You can refer those videos to understand how this metabolic reprogramming occurs in the pancreatic cells. So if you need to understand more about this cancer associated fibroblast and the stroma interaction, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.